What's up, guys? Welcome back. Today we are going to do the urinary system. Um, our main goals are to cover the anatomy and the histology a little bit, and then to discuss urinalysis. We start off with the basic structures. I'm going to draw you a picture of the functional unit called the nephron. Not the best picture, but it works to kind of describe to you how the kidney functions in taking blood, filtering it, reabsorbing certain components, and secreting certain components into the urine that we would like to get rid of. And then things that should not be in the urine or should be in the urine. So obviously we have a basic introduction here. Feel free to check that out. A couple keywords: filtration, reabsorption, and secretion. We'll discuss it. Here's the terms you need to know. As always, it's your list. Check it. Make sure you're familiar with everything and you can recognize them on models, real anatomy pictures, etc. Let's begin. First thing we have to have is blood flow. So clearly on here, you can see two kidneys. We've already discussed the adrenal glands and everything else. You have a kidney on the right and you have a kidney on the left. Some people only have one, but mostly people have two, which allows you to filter blood very efficiently. We have our adrenal glands, which we talked about. You should still know them. And we have these blood vessels. The two major blood vessels are your inferior vena cava, which is this blue guy here, big vein that drains all deoxygenated blood from the lower body upwards into the heart. And your aorta. Specifically in this region, this is called the abdominal aorta because it's in the abdomen. And this is the blood vessel that will connect to your renal arteries. So our renal arteries are the red blood vessels that deliver blood flow into the kidney. That's where it gets cleaned, if you want to call it that. And then the renal veins, left and right, left's only one labeled here, will take that clean blood and send it back into venous circulation. Important. After the kidney filters this blood, it turns it into urine. And we talked about this in the previous one. This urine comes down and it will be stored in the urinary bladder, which we can see here. And it drains from those kidneys to the bladder via the ureters. Obviously, you have one coming out of the left kidney and respectively coming out of the right kidney. Both of those drain into the urinary bladder where it is stored, and then when needed, it will be voided out via the urethra. So obviously on here, there's a uterus. So this is a female image. We already discussed that last lab. Now, on this model, we can see the kidneys, the adrenal glands. We can see the inferior vena cava and the abdominal aorta. We can see the renal artery going in on both sides. We could see the, I'll do that in blue, inferior vena cava. And we can see these renal veins coming out, as well as the ureters, which would come down, fill up the bladder, and your urethra would be coming out the bottom. Now let's take a look inside the kidney. I discussed that the renal artery, as you can see by the arrow, delivers red blood into the kidney. This is blood that needs to get filtered and cleaned. So there's three blood vessels I want you to know. This picture primarily is talking about the blood flow, as you could. How did I know that was going to happen? As we move from the renal artery down, right? Through right there. I'm primarily going to focus on some of these structures, and you will know which ones to focus on because I will show them to you. Obviously, blood comes in through the renal artery, then it branches off. The branching, these are called segmental arteries, right? So those are the first branches off of the renal artery. And then we have interlobar arteries. These arteries, and maybe I'll do it in different colors so you can see a little bit better, right? Renal comes in, branches off into segmental, and then it branches off into the interlobar. Those are ones that kind of go in between a structure I'm going to show you called the renal pyramids, and they will go out to the cortex of the kidney. So not only blood flow, right? And then obviously renal vein drains out respectively. We'll highlight that just so we don't forget. But there are a few important areas of the kidney. Now we're diving into it, right? Not just big superficial structures. The kidney is broken up into two generalized regions. The outer region is called the cortex, and I will highlight that for you here. The cortex is where we find a lot of these guys. This thing is a nephron, and that's the picture I'm going to draw, and we're going to talk about it because the nephron is the functional unit of the kidney. We have a million of them. They're super important, and that is the structure, group of structures that is doing the important stuff, doing the filtering, the reabsorbing, and the secretion, and thus forming our urine. The inner portion is called the renal medulla, and I'm going to erase this because I don't want to completely ruin the picture. But just like we saw with the adrenal gland, we have the cortex, and that medulla is towards the inside. Another structure here are these triangular shaped pyramids. They are called renal pyramids. They're extremely important. The nephrons project into these renal pyramids, and we'll talk about it. Obviously, once that urine is formed, it comes down and out through here. This area, you can't quite see it, but it's called the renal pelvis. I'll find a better picture for you. And then out into the ureter, right? So on here, because I just mentioned it, we have the renal pelvis and the ureter. Each of these models, you can kind of see something a little bit better than another model. Obviously, we have the renal artery and we have the renal vein. You can also see on here the segmental artery where it branches off. And you can kind of see the interlobar arteries over here. We can also see the renal pyramids. And this is a good time for me to introduce another component because it's on your list. And these are the calyx. This is a minor calyx, which I'm highlighting here for you. And then this is a major calyx. What these are structures that carry the urine once it is produced. So I'll try to maybe draw over this in a minute. The urine is produced kind of in that cortex. It drains down through here, through structures called collecting ducts, into the minor calyx, which collects the urine, through the major calyx, and then it dumps out into the renal pelvis, and then it obviously goes down the ureter into the bladder. So I will erase this because it looks kind of sloppy. So major and minor calyx are structures you should recognize, as well as the renal pyramids. Obviously out here, this is our cortex, and everything on the inside would be the renal medulla. This is a good picture to see those blood vessels. We can see the renal artery come in, segmental and interlobar arteries. We can see those renal pelvis, the renal pyramids, the renal pelvis, and the ureter. In addition, why I like this picture is we can see this green structure here I'm highlighting, and this is called a nephron. The kidney is stacked with a whole bunch of nephrons. That is important. That is the functional unit of the kidney. That is the thing doing the work. And I will draw you a picture of what that looks like. Obviously, you can see the cortex and the medulla on here as well. This is our nephron. It looks complicated. There's a lot of different parts. On here, instead of having it be spread out, it's kind of overlapped. That's how it is in the kidney because they're stacked up a bunch of them. You can see your major parts over here. You can see that minor calyx as well, which we were talking about. And you can see where this nephron lives. You can always reference this picture for the names and see how they're spelled and where everything is specifically located. But I'm going to draw you a simplified version of the nephron and introduce it like that. So the first thing I want to talk about is the nephron has blood flow coming into it and out of it. These will have names, right? This thing has a name. There is a collecting area. And then obviously after we go through this whole process, probably not orange, not the best idea, we should form a little bit of urine. So the main thing with this is I'll kind of tell you how it works a little bit, but also what the structures are, and then I will go show you on the model what we're labeling. So step number 1. This is called an afferent arteriole. And again, I'm going to abbreviate for space and time. But if you flip back one slide, you can see the name of all of these terms. I'll probably go back there and highlight that for you. These are the efferent arterioles. In between there, we have a structure called the glomerulus. These are also referred to as the glomerular capillaries. It's a very, very important structure, and I will tell you what it does after we label this. This is called the Bowman's capsule. This is called the PCT, which stands for proximal convoluted tubule. This structure right here is called the loop of Henle. It has 2 parts. It has a descending loop and an ascending loop. There are differences in the characteristics of these loops that I will discuss briefly, but way more in lecture. This is the distal convoluted tubule, or the DCT. This is the collecting duct. Obviously, whatever drains out of here will be urine. So those are the parts. What's happening during this process of urine formation is step number 1. Blood's coming in, and it gets filtered out through those capillaries. Capillaries are very small. These ones have small pores. So step number 1 will be filtration. Filtration is separating by size. Only small particles will be able to pass through. So what you have in here is what we call filtrate. Anything large stays in these blood vessels and will exit out through the efferent arteriole. Things that should not go into our nephron are large things like proteins, blood cells, red blood cells, white blood cells, and all that stuff. So if you find those in the urine, those would be abnormal findings. Step number 1, we filter the urine. All small stuff goes into the Bowman's capsule. Now there's stuff in there that we want to keep, like a lot of water, like glucose, like amino acids and ions. But there's some stuff that we want to get rid of. So as we progress through and this filtrate moves in, what we have is step 2, which is reabsorption. A lot of things get sucked back into the body. There are blood vessels that cover these structures, and as everything gets reabsorbed, for example, a lot of water, glucose, things like that, we pull it back into the body. Very, very important. A lot of water reabsorbed. We don't want to lose tons of water. We don't want to just pee all the time. And we definitely need that glucose and amino acids and some ions. Obviously, in lecture, we'll go through the specifics about how these things are transported, etc. So a lot of that happens in the PCT, or the proximal convoluted tubule. As we come down the loop of Henle, we reabsorb some more water. The ascending loop, let's get a little color here, is impermeable to water. So that loop of Henle, as we will discuss, is very important for creating concentrated urine, as well as there are hormones that work on that loop of Henle in the DCT. In the DCT, we do have a little bit more reabsorption, but the main thing I want to talk about is step 3, and that is secretion. We will secrete things like hydrogen ions, etc. We also have the exchange of other ions, sodium, potassium, things like that. And this allows us to get rid of some things that we might want to not keep, like acid and stuff. It also gives the urine a slightly acidic pH, which is very good, and we will talk about why. As this filtrate passes through, we have a little bit more reabsorption taking place over here in the collecting duct under the influence of ADH, which we have talked about in the past. And kind of our last chance, if you will, to reabsorb water. But what we end up with should be concentrated urine. Along this path, although it seems kind of complicated because it is, we filtered, reabsorbed what we needed to keep, secreted what we could afford to get rid of. But along the whole way, we just reabsorbed a lot of water. So that's important. So what we should have would be concentrated urine at the end with various components in it that we will discuss when we go into urinalysis, which is what is in the urine. That's your basic picture. You can look through here, right? You can see all these terms we talked about: Bowman's capsule, glomerulus, DCT, PCT, loop of Henle, collecting duct, right? So I'll show you where they are on the models. I like this picture. It shows the osmolarity and kind of what's happening as it goes through. It's also very simple. It's a nice place to practice your terminology. And here, this is the model. This is a picture of the renal cortex. So what we're looking at here is a nephron, and I'll label some of them for you. This would be our Bowman's capsule. This would be our PCT. This would be our loop of Henle, descending and ascending loop. This would be our DCT, distal convoluted tubule. And this would be our collecting duct. I will label another one for you. Not the best, but I'll try to show you. This would be our Bowman's capsule, PCT, descending loop, ascending loop, and then it's kind of weird here, but that would be your DCT, obviously draining into the collecting duct. Makes sense. The only thing I'm going to add in here is these are our glomerular capillaries, or our glomerulus, if you will. We have a bunch of nephrons. So each nephron, it doesn't show it here because it's just showing you the blood flow. Each nephron would be attaching to this. We'd have a Bowman's capsule and another squiggly guy over here, respectively. This is zooming in on what we call the renal corpuscle. The renal corpuscle is just the arteries coming in, the glomerulus, and that Bowman's capsule region. And this is important because we will see some histology slides that look like the one below. So we can see, and I'm going to draw an arrow over here so I don't close this thing out. We can see the afferent arteriole, right? We can see the efferent arteriole. These are the blood vessels here and here, respectively. We can see our glomerular capsule, or the glomerulus, if you will. And I'm going to stay away from that and get the Bowman's capsule, which is surrounding that. And then obviously all that dumps into the PCT. So I like this picture. You can kind of see what those capillaries look like. There's some other little structures in there I will mention. But this collectively is called the renal corpuscle. And this is our model. So we have coming in the afferent arteriole. These are the capillaries. We have a new structure here. These little spider-looking creatures. These are called podocytes. And I'll show you where they are here. Podocytes are the little cells that aid in that filtration process. They have little arms that reach around and kind of surround those capillaries. This is our Bowman's capsule. And this would be our PCT. Over here, just because I forgot to mention it, this would be our efferent arteriole. So blood's coming in like so, and it's leaving like so. Makes sense. There's a key. So all those models, if you want to go back and look at it, you can print this one out and take a look. It's a key that correlates to all those that I was showing you, because I didn't necessarily write them on there. I just highlighted them for you with the pen. You got it all. Now we'll do a little bit of histology. Not too much, just a little bit. The first one is the kidney, obviously. We can see the ureter right here, right? We can see that renal pelvis. We can see the difference between the cortex and the medulla. That's it, right? Basic picture of a kidney. This is zooming in on our renal corpuscles, and we'll see a couple pictures like this. Right off the bat, the first thing I recognize is that Bowman's capsule. Bowman's capsule. Coming off the Bowman's capsule will be the PCT. And what's inside the Bowman's capsule is your glomerulus. I'm not even going to do it because I'll do it like that. And this little afferent arteriole kind of coming in. Afferent arteriole coming in here. These are our capillaries. And it makes sense. This is obviously the artery that's delivering blood flow in to those afferent arterioles. It has a name, but I'm not discussing it here. So just looking at a couple Bowman's capsules. Another renal corpuscle. I will try to highlight yellow. The Bowman's capsule. Obviously, our PCT would be coming off over here. These are the capillaries that make up the glomerulus. And that's it. We have the ureter, which we've discussed as far as delivering that urine away from the kidney into the bladder. So this is a histology slide of that. We can see this epithelium on the inside. I'll mainly be able to recognize this slide as the ureter. You obviously have some muscularis or muscle, and then the adventia is towards the outside. So recognize this slide as the ureter. You can see it's kind of thick. The epithelium has certain characteristics that allow it to, stretch a little bit. This is a slide of the bladder. We can see that the bladder is made of transitional epithelium. And we have two slides on the urethras. We just saw that when we did reproduction, right? We know that the male urethra is surrounded by corpus spongiosum. We can see that it is very open on this picture, as opposed to the female urethra that is shaped slightly differently, has a little bit of muscle around there. So just recognize this slide as the female urethra. Male, female. We have some real anatomy pictures. Obviously, you'll go through all this stuff and you'll practice it and you'll do your homework and your study. We can see the same structures, the renal pyramids, the cortex, the medulla, these major calyx, right? That renal pelvis there, the arteries coming in, the veins coming out, the ureter. You should be familiar with it, right? Obviously, some of these pictures have the veins highlighted in blue and the arteries highlighted in red, which is pretty nice. There you go. And you have access to these. So check it out. You can see the difference between the major and minor calyx. The minors are the ones that are coming out here. The majors are the bigger ones draining out into that renal pelvis. Urinary bladder. Obviously, this is male because we can see the prostate. And now we get into urinalysis. So you know what things, you kind of have an idea of how urine's formed. Let us talk about what things are normally found in the urine and what things are not found in the urine that could be indicators of some type of problems. So our list, and I like this, obviously you have textbook, you could go through and read as well, but I like that this summarizes it. So first off, you get a sample of urine. The first thing you're going to recognize is differences in color. What do we want? I'll keep it red. What do we want the urine to look like? We want it to be pale yellow. It is because of the presence of uracil, right? We get a lot of nitrogenous waste in the urine. So how we get rid of it? And that would be considered normal urine. Obviously, hydration has a lot to do with color. If it's clear, that means you're super hydrated. I would still consider that normal. But pale yellow for most individuals would be a normal color. If it's not, you're going to have one of these, right? Yellow-orange could be an indicator that you have lots of bilirubin, possibly jaundice, maybe sickle cell, depending. And that the yellow-orange is very similar to the jaundice that we talked about with the presence of bilirubin. All the other ones, not good, right? That's not good either. But you have brown-green, red smoky, dark brown. Most of this has to do with the presence of hemoglobin or red blood cells or some type of crazy infection. So we will talk about the difference between hemoglobinuria and hematuria in a few minutes. But all these are clearly not good colors. It's not normal. Um, as far as obviously if it's green, right? I forgot the green. Probably have some type of bacterial infection. The transparency is the clarity. So when we look at urine, we want it to be clear. That's normal. If it's cloudy, very opaque, and sometimes whitish in color, that could be a good indicator of an infection, right? The pH, you don't have to worry about this. It's not really written properly. Um, the pH, normally we have it slightly acidic, right? Maybe 5, 6. 7 is neutral, it's okay. 8 and above, you start getting into very basic urine, and that could be an indicator of an infection. So our normal pH we'll say is between, let's say, 5 and 8. So I'll write that in for you there, right? That would be a normal pH. If we recall, acidity is a lower number, below 7. The lower the number, the more acidic. And the higher the number, the more basic, obviously, above 7. So 8 could be an indicator maybe the urine sat there for a little bit or possible infection. Specific gravity, I'll pick a different color, is how much solute you have in the urine. So simply put, there's a way we measure it. If you have 2 urine samples and one of these samples has a lot of stuff and the other sample has a little bit of stuff, this one has a higher specific gravity. This one has a lower specific gravity. A specific gravity of 1.000 is water. So a normal specific gravity is 1.001 to 1.03 on the lower end, aka that guy. Higher specific gravity with lots of stuff in the urine usually means your urine is more concentrated. It's been sitting there. You don't have a lot of water in it. It's a high concentration of stuff, of solute. So a high specific gravity above 1.03 is an indicator that you are dehydrated, right? You're conserving as much water as possible. There'll be a lot of stuff. The urine will usually be darker in the case of a higher specific gravity. Maybe you have proteins in there, aka just contributing to all that stuff in there. There could be several reasons for a high specific gravity. What does it smell like? It smells like urine, right? We call that an ammonia-like smell, which would be normal. 2 things we are not looking for, unless, of course, you're a ketogenic diet or something like that, would be a fruity acetone-like smell. This would be because of the presence of ketones. We find ketones present in diabetes mellitus, in times of starvation, sometimes low carbohydrate diets. But ideally, it does not smell super sweet acetone. It smells like ammonia, which would be regular urine. There are some things that we want. We want urea, right? And urochrome, which we talked about before as far as the pigmentation. We do not want to see protein in the urine. Protein in the urine could be an indicator to glomerulonephritis or some other conditions that we will discuss. It's not good. Protein should be too big to pass through those capillaries, and thus normal urine would not have protein in it. If we did have protein in it, it could be an indicator of some of those listed conditions, right? Do we want sugar in the urine? We do not want sugar in the urine. A normal sample of urine should be negative for glucose, which is obviously the sugar. If you have glucose in the urine, it is called glycosuria. You should be familiar with that, and we often see that with diabetes mellitus, right? In the case of diabetes mellitus, your blood sugar spikes super high. We cannot reabsorb all that sugar in the PCT like we talked about, and some of that sugar will pass by, and thus it will come out in the urine. So in a normal individual, non-diabetic, we should be able to reabsorb all that sugar because their blood sugar is maintained. But in the case of diabetes mellitus, we will often see glycosuria. Do we want ketones? Normally, no. If you have diabetes mellitus or you're on a low carbohydrate diet, or if you're in a state of starvation or ketosis, we would see ketonuria in the urine, and that's because the body's producing ketones and those will come out in the urine. Do we want to see blood in the urine or hemoglobin? No, we do not. That should be staying in the blood. Red blood cells are too big to pass through the capillaries. The hemoglobin should be in your red blood cells. So negative would be normal. If you do see, and I'll show you some situations, red urine, right? It means your red blood cells might be lysing. You have damage to those capillaries. You have some type of bleeding in the urinary tract, maybe menstrual cycle, or some type of infection, right? So clearly, bloody urine is not normal. And we already talked about this, but you do not want to see bilirubin in your urine either. If you see it, it is probably an indicator of hepatitis or some type of liver damage, obstruction of bile, and jaundice, which we have talked about several times before. SCA, which you'll see pop up a couple times, is sickle cell anemia. I just didn't describe that, but it's an abbreviation. It was on the previous slide as well. And urobilirubin and urochrome are normal. Sweet. So you should be familiar with the normal appearance, issues associated with it, and I'll show you some pictures. Specific gravity, what is it? I'll show you how we measure it. pH, and these are some conditions: the presence of protein, glucose, ketones, and red blood cells or hemoglobin are all not good. We want the urine to be, and we have some pictures just to follow up on the urinalysis. So we have some pictures. This is normal. It is pale yellow. It is pretty clear. This is pretty cloudy. It is usually an indicator of an infection. And it's not so good, even though the picture is blurry. That urine's cloudy. We have hydration in lecture when we talk about, various aspects of the urinary system. Water is important. Staying hydrated is important. Maintaining stable blood pressure and blood volumes is important. So if you start to see darker colored urine, that is an indicator that possibly you are dehydrated. The kidneys are saving water, so they are producing less volume of urine, and thus the volume you are producing would be a higher concentration, right? More stuff in there, less water, because you need to keep the water. So that urine would have a very high specific gravity, lots of stuff in it, in the case of dehydration. If you're looking at your pee and it's clearish or pale yellow, we could assume that you are well hydrated. Higher numbers, dehydrated. This is hematuria, the presence of blood cells in the urine. And this is hemoglobinuria. So I'll show you the difference between having red blood cells versus having hemoglobin. Now, hemoglobin is the protein found within red blood cells. So if your red blood cells are just present and we spin it, aka this is centrifuged, because we could see that description here, red blood cells are heavy, they sink to the bottom. Because both of these samples are red. How do we know the difference? If you have some type of disease, where your red blood cells are lysing open, and you have hemoglobin. When we centrifuge that, a little bit might go to the bottom, but for the most part, it stays suspended. So this is the difference between hematuria, which is the presence of red blood cells, and hemoglobinuria, which is the presence of hemoglobin. We centrifuge it. It's clearly red. Those cells don't sink to the bottom. Hemoglobinuria. This is normal. This is bilirubinuria. It said that the urine may take on a much darker appearance from the presence of bilirubin, oftentimes jaundice, things like that, liver issues will result in this very, very dark colored urine, as well as severe dehydration. This is a device called a hydrometer. What this picture is showing you is a hydrometer, but we use this to measure specific gravity. So you have that glass tube, you put the urine in it, you put this guy, and it floats, and there's little measurements on here. And it floats, and you can get a reading for how much solute is in the urine. Again, lower specific gravity, closer to 1.001, better. High specific gravity means you are, for some reason, there's more stuff. The urine is more concentrated. Lots of solutes dissolved in there, maybe presence of proteins and things like that. All not good. And traditionally, when you do urinalysis lab, you use a rapid screen, right? So you basically take your sample, you dip this thing in for a second, and it'll give you a readout. So you can find all of that data that I showed you for your analysis as far as normal or abnormal. You can get a quick scan based off a rapid screen test. Once you dip it, these little dots here change color, and then you would just match it up with the according chart that comes with every single test. So it would tell you, do you have the presence of leukocytes? What are your nitrates? Do you have protein, pH, right? Is there glucose present? What's the specific gravity, et cetera, et cetera, et cetera. And you would just match the colors. So, generalized overview. What did we cover? We covered the anatomy of the urinary system. We talked about the structures, and we talked about the blood flow going through. We also talked about the nephron, and we explained how the nephron kind of works in three aspects of filtration, reabsorption, and secretion, how we make concentrated urine with the good stuff in it and the bad stuff not in it. We showed some histology slides, and we talked about urinalysis. Urinalysis is the components that are in the urine, and you should be familiar with what should and should not be in there, as well as some characteristics associated if you do see some bad things in there. Other than that, that's your urinalysis lab. Obviously, we'll talk a lot about it in lecture, and obviously you have a test, so study for it. I will see you in the next one.